Prior to the very beginning, the beginning of our universe, time, space, and matter did not exist. Nothing. Then, Genesis 1:1 tells us, "In the beginning, God created the heavens and the earth." Since the dawn of creation, God's attributes have been seen and known by all. As a building requires a builder, creation demands a creator. An outstanding design points to a perfect designer. Our amazing universe reveals the glory of an awesome creator. God's wonders surround us, and these wonders reveal much about Him. Through creation, we get a glimpse of His power and His wisdom, His majesty and His care. Creation is speaking to those who will listen. God's incredible power is displayed everywhere, from the new growth of plants in the spring to new life forming in the womb. Wherever we turn, the Creator's energizing power is unmistakable. In this episode, we will shift our attention to the creation of life. The evidence we'll be looking at is by way of molecular biology, how the densest information storage mechanism known in the universe is found in our very DNA. We will explore how simple cells are infinitely complex in actuality, and most profoundly of all, how the probability of the formation of our cells in view of the natural process model is so infinitesimally small that the mind just cannot grasp it. As we will discover, matter independently does not, nor can it, give rise to information. Much less could it create an operating system that stores this information, reads it, and sends instructions to build and operate complex DNA strands of genetic code. From one single cell, much smaller than the tip of a pin, to your entire body with over 100 trillion various type cells which are themselves encoded with the same instruction manual. It's time to take a ride deep into the cellular level. Were we just incredibly lucky? Or was there more involved than blind chance and random interactions between matter and energy? Why is chance such an unsatisfying theory? Perhaps that many analysts have done the math and understand the formidable odds against success. Chance is defined as the unknown and unpredictable element in any event that has no assignable cause or guidance from a natural law or intelligent agent. Since the 17th century, one tool has been used consistently to predict the outcome of such events. It is called mathematical probability. In very broad strokes, here's how the theory works. When we flip a coin and call heads, we know that the probability is 50% or 1 chance out of 2 to be successful. Flip the coin twice and get 2 heads in a row gives us 1 chance out of 4. 3 flips with 3 heads on a single attempt gives us 1 out of 8. Every time an additional flip is added to continue getting heads, the chances are reduced exponentially by a multiple of 2. By the time we reach 10 flips in a row, our probability is reduced to 1 out of 1,024. We get the idea now. When applied to the origin of life on Earth and the random formation of large biomolecules, the probability theory clarifies that the limitations of chance is the creative agent on the primordial Earth. For example, what are the odds of a single protein could come exclusively through the blind interactions of chemistry? Our target is a smaller than average molecule made from 150 amino acids, each aligned to ensure a folded chain. Researchers have calculated the probability of success is 1 chance out of 10 to the 164th power. That's one correctly sequenced protein chain in 100 trillion, 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 and add another 7 trillions, failed attempts. That's 1 with 164 zeros. But despite these enormous odds, some theorists argue that, given enough time, anything is possible. So let's test the validity of this argument. First, we assume an ideal world with all the perfect conditions to provide every opportunity to succeed. We stock the oceans to capacity with amino acids. That means all the atoms on Earth, including its entire supply of carbon, nitrogen, oxygen, hydrogen, and sulfur, are present to form the 10 to the 41st complete sets of the 20 amino acids used to build proteins. Then we'll alter the laws of nature to protect these building blocks from the destructive rays of ultraviolet light and chemical contamination in this primordial soup. Now, let's turn this chemistry loose and see what happens. The amino acids start bonding furiously. An entire chain of 150 units self-assembles in only 1 second. Since all 20 types of acids are available at the majority of sites, there is a 5% or 1 out of 20 that the correct molecule will align on the chain. If the sequencing is incorrect, the chain is immediately destroyed and a new assembly begins. Throughout the planet, 6,000 million, billion, trillion attempts will take place every minute. That means in 4.6 billion years, the oldest estimated age of the Earth, the number of chains that don't fold will exceed 10 to the 58th power. It is a staggering total, but nowhere near 10 to the 164th power of trials necessary on average to build a protein with 150 amino acids by chance. Undirected chemistry cannot produce our coveted molecule during the entire history of the Earth. Scientists generally consider anything with a probability of less than 1 part in 10 to the 70th power operationally impossible. And that calculation does not take into consideration the information that's stored in the cell, which directs the cell with all aspects of operation. That estimate is solely the chance of chemicals combining to form into just something living. Now, suppose against all odds, chemical evolution produced our single protein. Would we have life? No. We'd simply only have one protein, a lifeless arrangement of amino acids. The simplest living cell that we know has over 300 different proteins. But proteins are only part of the story when we consider any actual cell. We will need carbohydrates, complex sugars, nucleic acids, DNA and RNA, lipids, and a whole variety of chemicals which jointly constitute the living state. Every one of those elements have to be brought into the same microenvironment and at the same moment in time. Each chemical building block must then be assembled and organized, the network of molecular machines that will control every facet of life. If we can appreciate just precisely how difficult it is to produce just one molecular machine using nothing except atoms and energy, we can then discover that we have a profound problem. Once we have one functioning molecular machine, you still don't have a living thing. These machines need other molecular machines to survive and replicate themselves. Even if nature was capable of producing all the molecular machines necessary, that still wouldn't be enough. They have to all be together inside this tiny little membrane bound in space that we call a cell. And it all has to come together at once. That means any of these machines were to miraculously be produced, they wouldn't survive long enough waiting for others to be produced. They would have to go on through the entire sequencing again and again until it was all created at once. This already appears mind-blowingly hopeless. Yet if you feel there's still hope, then let's continue. Everything inside the cell to live needs to work together in a causal loop. The higher level of organization transcends the pieces. The spatial organization in the cell requires that molecules end up in the right place and the right time. The DNA is copied into RNA, The polymerase that does the copying has to find the right location in the DNA to start copying. The RNA has to somehow hook up with each successive sequence that is perfectly coordinated. This is an awful lot to account for by random chance. The precise sequencing for proteins to form are determined by the precise arrangement of the bases in the DNA molecule. That means that this arrangement constitutes the code for the arrangement for the amino acids in the chain. Proteins without DNA could not exist, nor could DNA exist without the proteins. They have to be in existence together at the same time, and they had to be functioning within the cell at the very beginning of life. It could not have evolved because they require one another for sustenance and utility. They could have only been created simultaneously and instantaneously. Charles Darwin himself stated in his book On the Origin of Species that such structures could not hold up if it could be demonstrated that any complex organ or structure existed which could not possibly have been formed by numerous successive slight modifications. My theory would absolutely break down. Therefore, it should be stated that the naturalistic view on random processes and evolution is unscientific. With what we have just seen, extreme complex codes of information that could not have arisen out of pure chance, then the question is, does the complexity of DNA point to an intelligent designer? The code can only be compared to what we could see today in digital nanotechnology that directs all the proteins, sugars, amino acids, etc. in order to stay alive. Evolutionary biologist Dr. Richard Dawkins concedes that DNA is full of digital code, stating, "The machine code of the genes is uncannily computer-like." Bill Gates has said, "Human DNA is like a computer program, but far, far more advanced than any we've ever created." What are we essentially dealing with within this cell of information? To answer the question regarding the origin of life, we have to reconcile what the origin of information is. German scientist Bernd Olaf Kuppers himself stated that the problem of the origin of life is clearly basically equivalent to the problem of origin of biological information. If you want to give a computer a new function, we need to program it with a new code, provide it with new information. It is the same thing that is true in life. There's a connection between information and function. This DNA enigma is what has to be ultimately be solved. And we are looking at the very nature of what information is. We can then determine that, by definition, according to the Merriam-Webster dictionary, it is communication or reception of knowledge or intelligence. Knowledge obtained by instruction. The human genome has 3 billion characters of information in DNA. As outlined in my 2020 release, Better Than a Spear in the Head, we looked at the various types of evolution and that the only type that we can agree to is microevolution because this is the only one that's been observed. This is merely just adaptation within a living species to its environment along with the various characteristics that come about in its offspring. The problem with successive offspring from one generation to the next is that it loses information rather than introducing more to it. In 1957, Francis Crick, one of the scientists that discovered DNA strands inside the cell, proposed that chemicals called bases along the spine of the DNA molecule function as alphabetic characters in a written language or digital characters in a machine code. This language is what was responsible for directing protein synthesis. First, a large protein complex separates the tightly wound strands of the DNA to prepare it to be copied. During the process of transcription, the polymerase produces a single-stranded copy of the original instructions. This transcript then moves on into a molecular machine called a nuclear or complex, an information recognition device that controls the flow of information in and out of the cell's nucleus. This RNA of assembly instructions is then exported out of the cell through a special gateway called the nuclear pore complex, or NPC, and into the cytoplasm. It arrives at a two-part chemical factory, the site of a protein synthesis. This is where the process of translation begins. Once the construction of the chain is completed, it is transported into a barrel-shaped machine where it is folded into the precise shape required to perform its function. From there, it is released into the cytoplasm in order to do its job. It is all an extremely well-designed system with countless moving parts that relies on each other for functionality. One would have to be quite blind not to recognize that it all requires a designer, no different than expecting an intricately built mega machine like a fully loaded Maserati Levante GTS. Take away any one part from the engine and you have yourself a useless car that wouldn't even fire up. One final piece of the puzzle that needs to be looked at here is the bacterial flagellum. Any level of research on its many moving parts, you'll discover that this biological miracle contains a rotor, stator, driveshaft, motor, inner and outer membranes, hook, and filament that is essentially a self-propelled mechanism for travel through liquid for various functions, such as in females' reproductive tracts. The flagellum has all the essential parts of a propulsion system like that of an outboard motor. When you look at how the flagellum is assembled, you can clearly see a mechanical device with a rotor, a gear wheel, and a motor that turns and whips the tail to propel itself, a far more effective design than twisted air screw blades. In a term coined irreducible complexity, this device itself would have had to be fully functional because of its multi-component parts, all of which are required to function. If you remove one part, you lose the entire function of the system. As with the example mentioned within a woman's reproductive tract, this system would fail to reproduce, hence no further generations could arise to evolve any further. In the bacterial flagellum motor, there are a total of 40 different protein parts that is necessary for this machine to work. Put simply, it is impossible for the flagellum to have evolved because, as a perfect example of irreducible complexity, it could not have functioned without any other parts. They must have fully formed and fully functioning machines, and without these flagellum, biological structures could not survive no matter what the evolutionists claim. In no peer-reviewed secular paper will we find any claim that the type 3 secretion system, or the T3SS, could emerge through functional intermediate stages. In a mutant strain that scientists devised, no relic structures were seen in their findings, meaning that a confirmation was made that these structures cannot be assembled in intermediate stages or evolved. In Darwin's day, the cell was thought to be just a simple sac filled with jellied carbon. However, after 150 years of cell research, we've discovered that they are hugely complex. Many thousands of different operations are taking place continuously within each of the cell's main parts, called organelles. Thus, the cell cannot live until all these parts are working properly. Even the simple E. coli bacterium has 4,000 genes. If we were able to magnify the DNA of the E. coli to the thickness of a clothesline, it would be 5 miles long. Therefore, there's no such thing as a simple cell. In conclusion, our living cells could not live without all these thousands of parts, shows that the cells were created in their finished form, just as the Bible says in Genesis 1. Even bacteria declare the glory of our Creator. Join me next time in part 2 of this Creation of Life series, where we will continue with DNA and the miracle of the cell. Here on Evidence Uncovered.